Okay, we're continuing with this book here, The Cancer Resolution by Mark Lintern. We're now going to go through page 130 to page 145. Okay, so first thing he talks about is, and I have the page numbers on here for anybody who wants to buy the book and go through it. It's like one of the best books ever written about cancer. The release of lactate. Lactic acid, when it gives off its proton, it becomes lactate, and then it has the proton, which is a manifestation of acid as well. And they both get pumped into the extracellular space when the cancer cell is running glycolysis, because it can't allow that accumulation of acid because it would kill the cell. So it has to pump it into the extracellular space. But that accumulation of acid in the extracellular space has negative consequences. It suppresses the immune system. Okay, it also contributes to activation of angiogenesis, the ingrowth of blood vessels that facilitate the cancer spreading to other places, to metastasis. Okay, so release of lactate contributed to in vivo development. In vivo means live development of tumor vasculature. Okay, it is well known that lactate is a key player in angiogenesis. Angio means blood vessel, genesis is to start something, to grow something. So it means the growth of blood vessels. Stimulation of VEGF, vascular endothelial growth factor. Okay, this is the growth pathway. Wound healing and repair. You're going to have VEGF and ingrowth of new vessels and wound healing and repair of tissue because they're going to be hypoxic. Whenever you injure a tissue, you have more cells that are far from the capillary. Whenever you have inflammation, due to the influx of fluid and immune cells, you're going to have increased distance from capillary to the regular cells, so they're going to be relatively hypoxic. In cancer, lactate plays an important role in angiogenesis, stimulating VEGF expression in endothelial cells. Those are the lining cells of arteries. Tissue hypoxia stimulates HIF, hypoxia-inducible factor, and this leads to VEGF production. Okay, so basically, hypoxia in animals, traditionally, the main cause of hypoxia was an infection and inflammation, okay? But nowadays, in modern atherosclerotic humans, it's from atherosclerosis, diabetes, hypertension, advanced glycation end products, thickening of the capillary basement membrane, all that sort of stuff. Damage to collagen from all the things I've talked about too contributes to that. Okay, so what I'm saying here is anything that causes hypoxia is going to increase the chance that this cell could revert to a primitive form, like an anaerobic bacteria, if you will, and also simultaneously activate the angiogenesis pathway. This is what cells do when they're threatened by hypoxia. They know that if that hypoxia persists and becomes worse, they're going to die. So they switch to anaerobic metabolism so they can function without oxygen, and then they start releasing these chemicals like VEGF to stimulate the growth of blood vessels so they can get closer to a blood vessel to give them oxygen so they don't die. So it all makes sense. You know, in nature, you don't have atherosclerosis. When humans are eating plant-based diets in the wild, they don't ever get atherosclerosis. This is a modern thing. Okay, culturing cancer cells in even a slightly acidic medium causes dramatic increase in both cell migration and cell invasion of other tissues. It has been known for over 3 decades, over 30 years, that elevated lactate level is highly correlated with metastasis in different forms of cancer. When a human cell's mitochondria have a severe injury, this can cause the cell to change to a primitive, powerful stem cell, what is called a mesenchymal cell. Okay? And a cancer of an epithelial cell is called a carcinoma. And that's what the majority of cancer is. The majority of cancers that have the potential to kill the patient, rabbits, so to speak. And they'll undergo something called an EMT, an epithelial to mesenchymal cell transition. And what they mean by mesenchymal cell, to revert to becoming like a stem cell. So what they're saying here is the cancer cell isn't just becoming like an anaerobic bacteria in this case. It's becoming worse than that. It's becoming like a stem cell. What's unique about a stem cell, it'll both have immortality. Unlike, other cells have a Hayflick limit, meaning they only get 60 cell divisions and they die. So it'll have unlimited cell division. That's called immortality for a cell. And it'll also have what is called pluripotentiality. It'll potentially have that. So the cell is developing more ability to change in different things. Because a normal cell in an organ can't do that. A brain cell is a brain cell, and that's it till it dies. So this is called de-differentiation. So those brain cells are differentiated. They're highly differentiated, your brain neurons, versus a cell changing from an epithelial cell to a mesenchymal cell where it becomes a stem cell. That's called de-differentiation. It has lost its specialization. It's no longer differentiated. It's no longer specialized. It is de-differentiated. And that's a sign of an aggressive tumor. They'll say this is a highly de-differentiated tumor with extensive angiogenesis. So that's really bad. It's reverted to being stem cell-like with lots of blood vessels ingrowing through which it can metastasize all over the place. So this is one potential step in the path to becoming an aggressive metastatic cancer. EMT, an epithelial to mesenchymal transition, okay, to becoming a cancer stem cell. And it's thought that this sort of transformation back to a stem cell is related to an animal's response to severe injury, like an amphibian, which can regrow its tail or even an entire limb. So we talked about how a cell can revert to becoming like an anaerobic bacteria, simply wants to grow. But what they're talking about here with an EMT is that it not only wants to grow, but it can grow a variety of things. So that's epithelial to mesenchymal cell transition. You're going to hear that word again, so it's good to know it. In other words, the cell is not just turning it into an anaerobic bacteria, but rather into a stem cell, or like an anaerobic glycolysis running stem cell for growth. Because when a cell is in a growth pattern, it runs glycolysis. What I'm getting at is, let's say you have a normal cell in an embryo or in a child. When a cell is in the phase of rapid growth, it doesn't need a lot of ATP to grow. It just needs a lot of ATP when it's a worker. So when it's in a growth phase, it'll run on anaerobic glycolysis, and it'll use those carbon skeletons coming into it in the form of glucose, glutamine, and fats to make the molecules, because the cell has to make a copy of itself before it can replicate. So the point is, to be a worker cell, like a normal, highly differentiated liver cell in an adult, you need tons of ATP. So the focus is on oxidative metabolism, which makes 18 times more ATP than does anaerobic metabolism, so the cell can be a good worker. Versus once you've reverted to this anaerobic form of metabolism, you switch on the growth pathways simultaneously, and that doesn't require much ATP. And in a sense, the cell is almost like trying to grow its way out of its hypoxic death zone. Okay, he next says that this ability of chemo to damage the mitochondria in a cell and cause it to undergo epithelial to mesenchymal transition is part of what can explain how chemotherapy can make the cancer worse. It might shrink the tumor size initially, but it can change the behavior pattern of the stem cells within the tumor and make them more aggressive, more of a epithelial cell to mesenchymal cell transition, making them have more de-differentiation, more pluripotentiality, more aggressive growth. Okay? The Warburg effect is the most important hallmark of cancer. That's a big statement right there. The Warburg effect is the most important hallmark of cancer. So that's, again, the idea of some form of mitochondrial injury with the cell reverting to metabolism by glycolysis, anaerobic metabolism, essentially. Okay, but that can occur in the presence of oxygen, even when the oxygen is brought back. What causes metabolic acidosis? Well, for example, animal protein, because there's more sulfur side groups, which are in part metabolized to sulfuric acid. Excess sodium chloride, because the chloride being an anion, negatively charged ion, will displace the bicarbonate, and that can lead to low-grade metabolic acidosis. Lack of alkaline foods, a lack of fruits and vegetables. Okay, what causes tissue hypoxia? High-fat diets, because it sticks the red blood cells together. It can also cause glycocalyx shedding and damage the endothelium. It also causes hypertension. It also causes insulin resistance and diabetes. Amyloid clotting from leaky gut, or amyloid clotting due to iron overload with free iron in the blood, leading to ferrous redox cycling. Glutamine fermentation is something that cancer cells can do. They can make a little bit of ATP from glutamine inside the mitochondria. So they call this substrate level phosphorylation, SLP. And since it happens in the mitochondria, they'll call it MSLP, mitochondrial substrate level phosphorylation. And this is another reminder that the mitochondria has some ability to still function. Professor Michael Asanti, he's this great cancer researcher, he said that he showed from his experience that ketones can fuel tumor growth. And that's another big statement, because previously some researchers like Professor Thomas Seyfried had thought that ketogenic diet might be a good way to treat cancers. But What Lasante's saying is no, it's not. Cancer can feed on tumor growth. He says it's happy to feed on ketones. Lasante showed in breast cancer, for example, that there's some activity in mitochondrial oxphos and TCA cycles. Okay, he's pretty famous, Michael Lasante, for the idea of the reverse Warburg effect. So we'll talk about all that stuff later. That's it for today. I hope you found it helpful. Again, the page numbers are all on here if you're interested for the book.